I'm glad to see so many of you here today on this very special day for me. And the title of my PhD thesis is The Properties of Hydrated Nanocellulose Network Structures. And the most interesting aspect of this work is actually the solidification of water in fibrillar hydrogels, which is a remarkable transition of water from a free-flowing state into a solid-like network structure. And by incorporating a tiny amount of nanocellulose particles, we can actually create hydrogel network structures containing up to 99.9% of water with a self-supporting structure holding the water in place. And in this work, we refer to these materials as solidified water. And then we can ask ourselves, how is this even possible? And I would say two things. The properties of our chemically modified cellulose nanofibrils in terms of their dimension, which allow for particle entanglements at low concentrations, as well as the surface charge density, which is responsible for the colloidal stability in our liquid dispersion, as well as the particle stiffness, which will provide stability and strength to our network structure. But equally important is the interactions we have in our fibrillar hydrogels. And there we can have everything from double layer repulsion to our attractive van der Waals forces and entanglements. And the outline of today's presentation is that I will go through some relevant background for this research topic, and then I will go through the objectives we have stated in this work before I move on to the result and the discussion part, which I have divided into 4 sections. And the first part will cover how the fibril characteristics influences the dispersion properties. And then I will cover the experimental and theoretical description of isotropic hydrogels in the second part. And in the third part, we will focus on anisotropic hydrogels. And then in the final part, we will actually look at how can we functionalize both wet and dry CNF network structures to add new properties to these materials. And then in the end, I will also state the conclusions we have drawn from this work. So in this work, we have entirely focused on wood-derived cellulose nanofibrils. And the cellulose nanofibrils in wood are embedded in a matrix of hemicellulose and lignin in the cell wall. And by carefully remove the hemicellulose and lignin using chemical reactions and mechanical disintegration, we can obtain cellulose nanofibrils with widths ranging from 2 to 5 nanometers and lengths over 500 nanometers. This means that we will have particles with high aspect ratio, and the aspect ratio is the length divided with the width. And we will also have particles with good mechanical properties. And maybe the most important property for this work is actually that these materials exhibit complex rheological behavior already at low particle concentrations. So when we have our cellulose nanofibrils, we can actually disperse them in water and create colloidally stable CNF dispersions that are transparent. We can then further process these dispersions and create an isotropic hydrogels. And we create isotropic hydrogels by reducing the electrostatic repulsion between the particles, allowing the particles to come closer together and form a uniformly distributed hydrogel network structure. But we can also create anisotropic hydrogels. If we vacuum filtrate the dispersion and gently dries it and then reswells it in water or in an electrolyte solution. And what makes the anisotropic hydrogel special is that the fibrils are aligned into planes, which allows for them to swell in one direction. If we prepare our CNF network structure at higher concentration of fibrils, we can instead create aerogels if we freeze the structure, cross-links it, and then gently dries it. And in this work, this is the 3 different types of CNF network structures we have studied. And this leads us to the objectives of this thesis. And the overall aim of this work was that we want an increased understanding of the properties of CNF dispersions and gels and how the relation to their network structure. And to achieve this, we stated the following objectives. We want to understand how the fibril characteristics influences the dispersion properties, as well as we want to characterize network structure of the hydrogel and its mechanics. And we also want to identify which factors controlling the network structure and the formation of contact points. And finally, we also wanted to functionalize both the hydrogels and aerogels in order to add new properties to these materials. And by achieving these objectives, we're actually working towards these 3 UN goals. So this leads us to the first part of the result and the discussion, which covers the fibrils characteristics in relation to dispersion properties. So in this work, we have worked with a wide variety of different fibrils, and we have put a lot of effort in characterizing them in detail. And in this work, we have prepared tempo-oxidized fibrils, carboxymethylated fibrils, and coordinized fibrils. And in tempo-oxidation and carboxymethylation, you obtain fibrils with a negative surface charge. And using the quaternization reaction, we instead get positively charged fibrils. And by changing the chemical reactions, we can also prepare fibrils with different charge densities. And in this work, we have charge densities ranging from 400 to 1100 micromoles per gram. And we have also prepared fibrils with different aspect ratio by varying the raw material and the processing conditions. So this leads us to how the fibril characteristics influence the dispersion properties. So then we prepared dispersion from our fibrils, and we wanted to characterize them in the wet state, and to do that we used Sachs characterization. And from the Sachs characterization, we can see that we obtain a fibril width around 2.5 nanometer, which is very consistent with what we see in AFM. We can also see that if we increase the charge density of our particles, we increases the degree of fibrillation, as well as if we change from anionic fibrils to cationic fibrils, or use fibrils with higher aspect ratio, this will also increase the degree of fibrillation. And to better understand the flow behavior of our particles in the dispersion, we actually characterize them using rheo-optical flow stop. And in this measurement, we are flowing the dispersion through a flow cell, and we can determine the Brownian motion. And from the Brownian motion, we can actually determine the hydrodynamic length for our fibrils. And if we do that, we can see that if we increase the particle concentration of fibrils, we will increase the hydrodynamic length. And the most likely explanation for that is if we have more fibrils, they can entangle more, which will influence the motion of the fibrils. We can also see that if we increase the charge density, we decreases the hydrodynamic length. And this is most likely a result of increased repulsion, which can prevent some entanglements in the liquid dispersion. We can also see that if we increase the aspect ratio, we increases the hydrodynamic length. But now, if we have longer particles, of course, they can entangle more in the liquid dispersion. And this leads us to the second part, in which we will talk about how we can experimentally and theoretically describe the isotropic hydrogels. So in this work, we're very interested in the low concentration of fibrils we can use to actually prepare these hydrogel network structures. So in this work, we actually try to determine what is the lowest possible concentration we can use and still form a hydrogel network structure. And as you can see here, in this work, we managed to actually create a hydrogel with a concentration of 0.1 gram per liter of CNF and still have a self-supporting structure. And if we compare our work with other relevant studies, we can see that we have actually pushed the limit of the lowest concentration we can use to create self-supporting hydrogels and characterize their mechanical properties. But in order to do that, we actually needed to prepare a very special sample preparation, which allowed us to actually characterize the undisturbed network structure. So to do that, we actually designed a PDMS mold, which we can disassemble into 2 pieces. So this allows us to create the hydrogel before the measurement and make sure it can fully diffuse, and then move it to the rheometer and measure the rheological properties. And then so this method actually allowed us to measure the rheological properties of many different types of hydrogels. And from the frequency sweep, we can actually get insight about the stiffness of our hydrogel network structure. And here you can see that if we increase the concentration of fibrils in our hydrogel network structure, we also increases the storage modulus. But this is not so surprising, because if we increase the concentration, we will have more fibrils in the system, which can form more contact points. We can also see here that if we increase the charge density, we decreases the hydrogel stiffness. And I think this is most likely related to that increased charge density will change the fibril width, which will change the bending stiffness of the particles. We can also see that if we increase the aspect ratio of our particles, we increases the hydrogel stiffness. And this is most likely related to that higher aspect ratio particles can form more contact points in our hydrogel network structure. If we change the chemical modification from anionic to cationic fibrils, we see that the storage modulus is reduced. And this is most likely related to that the cationic fibrils can repel each other more than the anionic fibrils. And we can also look at how these hydrogels respond upon deformation. And here we compared low aspect ratio fibrils with high aspect ratio fibrils in order to see how does the material respond upon an applied stress. And what we can see is that higher aspect ratio managed to endure a larger number of stress relaxation cycles, as well as we have lower strains, which means that the high aspect ratio samples can resist deformation better than the low aspect ratio fibrils. But we also wanted to better understand the network topology of our network structure. And to do that, we actually, together with Stefan Lindström at Mid-Sweden University, developed theoretical network models we can use to get a better understanding of this. And in this model, we say that if we study how the storage modulus scales against the volume fraction, we can actually understand how our network structure is formed. So in this model, we have two case scenarios. And in the first case, we say that if we have a fibrillar system at higher concentration, the system will be very crowded and the fibrils will be restricted in motion. And this will not allow them to seek its contact points during the network formation. And in that sense, we say that the fibrillar structure is formed in a random way. And if this is the case, we say that the storage modulus should scale against the volume fraction to the power of 4. However, if we are in a more diluted system, the fibrils should have like the time and the freedom and the space to actually move around and seek its contact points during the network formation. And this we consider that the fibrillar network structure is then more formed in a more organized way. And if this is the case, we say that the storage modulus should scale against the volume fraction to the power of 2. And if we compare our results, we can see that we are much closer to the power of 2 than 4, which we take as an indication that we are forming our network structure with this more organized mechanism. But we also wanted to get a better understanding of the network mechanics. And to do that, we actually used Evans' model to describe our fibrillar network structure. So here we say that our CNF network structure and its contact points we can consider as an even model with a number of Jenkins elements coupled in parallel. And in this model, we have two deformation modes. We have the first deformation, which is an elastic deformation, which corresponds to that the fibrils can bending. We also have a plastic deformation, which corresponds to the fibril contact points can slide up on an applied stress. However, in a CNF network structure, we have to remember that we will have a wide variety of different interactions contributing in the network structure. And to compensate for this, we actually needed to employ an exponential distribution of the yield stresses. And this means that this is the equation we end up with to describe our network structure. But still, one question remains, because in this model, we're assuming that the deformation mechanism is that the fibril contact points are sliding up on a deformation. So to test this, we actually did a cyclic creep test, but with an intermediate time sweep in order to see how the storage modulus evolves after loading and unloading the sample. And as you can see here, for us, the storage modulus remains quite constant after loading and unloading the sample, which is an indication that our fibrils are sliding up on an applied stress, and once the stress is released, the contact points can reform. However, if we would have a permanent debonding, we would expect the storage modulus to dramatically decrease after each loading and unloading of the sample. So now we know we can actually use this model to predict the shear stress of our sample. So what we did then was that we took the maximum strain at each stress and we fitted this into Ivan's model. And then we could actually predict the shear stress for our different hydrogels. And as you can see, we have a quite good agreement for almost all samples, which is an indication that this model works quite well when we compare the shear stress with the experimentally storage modulus for hydrogels measured at rest. So this brings us to the third part, which will cover experimental and theoretical description of the anisotropic hydrogels. And as you remember from the beginning, what makes the anisotropic hydrogel special is that they can swell in one direction, which means that they have unidirectional properties. But in these systems, we also still have the charges present, which makes them special. And this is why they can swell. And to test how then the swelling pressure is related to fibril properties as well as electrolyte concentration, we actually prepared different anisotropic hydrogels. We reswell them in different electrolyte concentrations, and then we could actually see how the swelling pressure was related to these different properties. So what we could see that for charged fibrils, the swelling pressure is very related to the electrolyte concentration. However, if we look at the hollow fibrils, which only have the charge from the remaining hemicellulose on the surface, we could see that they were very independent on the electrolyte concentration. And the most likely explanation for this is that the hemicellulose on the surface can entangle, which will reduce the effect of the ionic pressure. And to describe these network structures, we again developed an elastoplastic model. And we still, in this model, have our elastic deformation corresponding to the bending of our fibrils, and we still also have the plastic deformation corresponding to the sliding of contact points. However, now, since we still have the charges in our system, we also need to have an elastic deformation corresponding to the osmotic pressure. And if we adapt our data into this model, we can see that we can get values of the elastic modulus ranging from 100 to 300, which kilopascals, which is relatively low given the high solidity we have in these network structures. But this just shows the anisotropy of these networks. So then we also wanted to understand how the mechanical properties of our anisotropic hydrogels are influenced by the aspect ratio. And to test this, we actually prepared anisotropic hydrogels with different aspect ratios, and then we did tensile testing. And what we could see here is that if we decreases the aspect ratio, we increases both the wet stiffness and strength of our network structure. And this is most likely a result of how the fibrils can pack in the network structure, because lower aspect ratio fibrils can pack more densely, which will give stronger hydrogel network structure. And to better understand the network mechanics from these materials, we actually started to use the beam bending model. So if we adapted our data to the beam bending model, we could see that this is a quite good fit, and we can use this model to describe the network structure at higher concentration of fibrils. What we also can see in the beam bending model is that it doesn't really matter which multivalent ion we use to create a hydrogel, because we see they end up more or less in the same place in the graph, which is an indication that they do not really influence the bond strength in the contact points. And we could also see that the aspect ratio will influence the topology of the network structure. So this leads us to the last part of the result and the discussion, which is about functionalization of wet and dry CNF network structures. So now we have seen that we can create hydrogels with very low concentrations of fibrils and still have very decent mechanical properties. So we wanted to fully exploit these materials by exploring the possibility of creating flow channels within the hydrogel. So as you can see here, we created a flow channel within the hydrogel by forming the hydrogel around a cylindrical body, which we carefully could remove from the hydrogel, leaving an open channel inside the hydrogel. And we can also see here that we actually also have the possibility of creating flow channels crossing each other inside the hydrogel, allowing for fluid mixing inside the hydrogel. And we could also actually use the layer by layer in order to add new functionalities to these channels. So in this image, you can actually see how we adsorbed from the QCM images, how we adsorbed PIH and MMT clay in order to create a layer by layer coating inside the hydrogel. So now we have looked at how we can functionalize wet CNF network structures. So therefore, we also wanted to see, can we prepare dry CNF network structures at higher concentrations of fibrils and functionalize them to add new properties to these materials? And to do that, we prepared aerogels, and then we used the layer by layer technique to create different surface coverage of silica particles on the pore wall in the aerogel. And these porous materials can be used, for example, in absorption materials. So therefore, we were interested in see how does the particle modification influences the liquid spreading of the CNF network structure. So to test this, we actually developed a method which allowed us to prepare tubular-shaped CNF aerogels, which we could gently move towards the water reservoir. And as soon as the aerogel hits the water, the liquid spreading can start in the horizontal direction. And by recording this entire event with a high-speed camera, we could actually determine the spreading length as a function of time. And if we did that and analyzed our data, we can see that the increased number of bilayers in the particle modification increases the liquid spreading rate. But we also wanted to compare our data to the very classical Lucas-Washburn equation, which is used to describe the liquid spreading in porous materials. And from the Lucas-Washburn equation, then you can determine the average radius of the pore size in your network structure. And when we did that, we could see that for our samples, we get a pore radius around from 3 micrometers up to 5 micrometers. However, from the SEM images, we know that this is much lower than the actual pore size. And therefore, we realized that we needed another expression to describe the liquid spreading, which also takes into account the surface area of the sample. And in order to do that, we actually combined the Dunstan and White's together with Kosenik-Karman's equation to get an expression that describes the liquid spreading as a function of time, but also considers the specific surface area. And if we compare our results by adapting our data to this expression with the theoretical surface area, we see that we have a quite good agreement for the results, which is an indication that this is a good way of determining the surface area of our porous structures. So this leads us to the conclusions of this work. So in this work, we have shown that we can create hydrogel network structures containing as much as 99.99% of water with a self-supporting structure holding the water in place. We have also developed theoretical networks models, which we can use to describe the network topology of our network structures, as well as understanding the swelling properties and the network mechanics in our network structure. We have also learned that the deformation mechanism in these CNF networks is the sliding of fibril contact points up on applied stress. And we have also learned that the aspect ratio for the particles is very important for the hydrogel formation, since it will determine the number of contact points we have in the system, as well as it will determine the initial topology. And then we have also seen that we can create flow channels within the network structure, as well as layer-by-layer modification is a very useful method to add new functionalities to CNF network structures. So that leads me to the acknowledgement, and then I would first like to express my sincere gratitude to Lars Wågberg for all support during this year, as well as Tobias Benselfeld, who has been very important for this work. Also Stefan Lindström, who has been very, very helpful in helping us describing our hydrogel network structure with these theoretical models, as well as Knut and Alice Wallenberg Foundation through the Wallenberg Wood Science Center for funding my PhD project, and of course the Fiber Technology Group and my co-authors.